Welcome to this video of pathogenesis of type 2 diabetes mellitus. I won't be telling in depth, right? I'll be telling only those things which are actually important for your clinical practice. So first, insulin synthesis. If you take anything in diabetes, it starts with insulin and it ends with insulin. That's why let's start with insulin. So as you all know, insulin is secreted from the beta cells of the pancreas. How is it? So initially it is synthesized as pre-proinsulin. Now what happens? This pre-proinsulin loses one peptide and becomes proinsulin. Now this proinsulin loses one more C peptide, like it's a C peptide. This is very important. And then it becomes insulin. So whenever our body produces insulin, it produces C peptide also. What is the importance of the C peptide? You can use it to understand whether the body is producing sufficient insulin or not. Okay. Now take the first scenario. There is increased blood glucose, but when you measured C peptide levels, the C peptide levels were very low. So it means that the blood glucose levels are high because the body is not able to synthesize sufficient amount of insulin. This is mostly in type 1 diabetes mellitus. Now let's take another scenario. So in this scenario also the patient has high levels of blood glucose. So you measured C peptide levels and you saw that the C peptide levels are normal or increased. What does it say? It says that the body is producing sufficient amount of insulin, but this insulin is unable to act on the target cells, which means that there is insulin resistance, mostly type 2. Okay, so proinsulin releases C peptide and becomes insulin. This C peptide can be useful in assessing the body's ability to secrete insulin. Now, the next important thing that you need to understand is the fasting and feeding cycle. So first let's understand the fasting cycle. What happens in fasting? The person is not taking glucose or food, there is low blood glucose levels. We all know that the important function of insulin is to reduce the blood glucose levels. When the blood glucose is already less, do you think there is a role of insulin? No, right? Therefore, the insulin levels are low, but the body now is trying to increase the glucose levels. Therefore, the glucagon levels are more. In fasting, there is low blood glucose. Therefore, the insulin levels are low, but glucagon levels are high. Now, what is happening? The body is trying to increase body glucose. It causes like splitting of glycogen, like glycogenolysis, and it increases hepatic gluconeogenesis. And what it also does, it decreases the glucose uptake by skeletal muscles, which ultimately maintains the blood glucose levels. Okay. Now, what happens in feeding cycle? Let's see. Whenever a person has food, the blood levels of glucose increase. Now there is the role of insulin. Now because the blood glucose levels are high, now insulin is very happy now. It comes to the blood circulation. There is increased insulin and decreased glucagon. This is very important. In healthy individuals, postprandially, the glucagon levels are low and insulin levels are high. What this insulin does now? This insulin decreases the glycogenolysis and it decreases the hepatic gluconeogenesis. Then this insulin also increases the glucose uptake by skeletal muscles, which ultimately lowers the blood glucose levels. So the important thing is, in fasting, insulin levels are low, glucagon levels are high, and after food like feeding, the insulin levels are high and glucagon levels are low. In the previous slide, we understood that whenever the glucose levels are high, insulin is secreted. Now, how this secretion actually occurs? So, if this is the blood vessel, after food, glucose levels increase. Now, this glucose is actually entering into the beta cell of this is the beta cell of the pancreas, this whole one. Okay. Now, so this glucose is actually entering into the blood glucose. This glucose is getting converted into glucose 6 phosphate by the enzyme glucokinase. So to trigger insulin release, this is very important. Glucose 6 phosphate is very important. Now this glucose 6 phosphate ultimately enters the Krebs cycle and it releases too many ATPs. Okay. You need energy to close a channel also. Therefore, what these ATPs do, these ATPs actually close these potassium channels. These potassiums were actually happily going outside the beta cells, but now what happened? This channel, this gate is blocked. Therefore, now the potassium is getting accumulated inside the cell. What happens when a positive ion gets accumulated inside the cell? The cell goes into depolarization. There is depolarization and there is calcium influx. This calcium influx will trigger the insulin release. So this is how the insulin released. So we'll quickly recap. The glucose was there. Glucose entered into the beta cell by GLUT2 receptor. This glucose was converted into glucose 6 phosphate, the triggering molecule. Glucose 6 phosphate ultimately led to formation of ATPs. These ATPs closed potassium channels, leading to depolarization, calcium influx and insulin release. Okay. Now, core causes. If you ask me, right, how do you say what are the core causes? I would just tell there are two core causes. So one is the genetics and other is the obesity. We all know, but let's understand how these things actually cause diabetes. So first one, genetics. What does genetics do? Genetics can impair any gene, right? It has that ability. So genes, it can impair genes which are responsible for insulin synthesis and cause decreased insulin secretion. It can impair insulin receptors also and it can cause insulin resistance. Now, how does obesity actually cause insulin, like diabetes mellitus? What happens, see, obesity means there is lot of fat inside the body. Now, what these adipocytes do, these adipocytes start accumulating fat inside it and they grow in size. So, in obesity, because of accumulation of fat, there is increased size of adipocytes. Now these adipocytes, what they do, they start releasing inflammatory markers. There is increased free fatty acids and increased inflammatory markers like tumor necrosis factor alpha and interleukins. There is a state of inflammation here. So now this inflammatory state, what it does, it actually starts altering the insulin receptors. Apart from that, the fat not only gets accumulated in the adipocytes, the fat also starts getting accumulated inside the muscles. So now when muscles were lean, right? So when muscles were lean, they were functioning properly. But now the muscles are also getting accumulated with fat. What happens? There is impaired insulin receptor signaling. So now if the insulin says, hey muscle, take this glucose from blood, the skeletal muscle is like, I already have fat, dude, like I can't take glucose anymore inside me. So this is happening. So which ultimately, because of this inflammation, because of this impaired insulin receptor signaling, there is a decreased response to insulin, leading to insulin resistance. So let's understand what happens in diabetes mellitus. Right, this is the blood vessel, and whenever a person has food, right, the glucose levels in the blood increase. Now what happens when glucose, like we read feeding cycle previously? So there is insulin released by the pancreas, right? Now what does this insulin? This insulin will ensure that this glucose is taken up by the muscle and this glucose is taken up by the liver, right? The excess glucose is taken up by the muscle and the liver, and therefore now what happens? The glucose levels are balanced. Now it normalizes. Now what happens in diabetes mellitus? So in diabetes mellitus, this uptake is impaired. Now even though the insulin is saying take this glucose, the muscle is not able to take this glucose, the liver is not able to take this glucose. Okay. Now the liver is unable to get glucose from the blood. The liver doesn't understand that this is a receptor problem. The liver thinks that there is no enough glucose in the blood and that's why I'm not getting glucose. Therefore, now the liver starts producing glucose. There is hepatic gluconeogenesis. This further increases the glucose. Can you see now how the glucose levels are increased? Now, diabetes is like, it's like there is lot of resources, there is lot of food, but still there is famine or drought. Okay, same thing happens. The blood is filled with glucose, but cells are unable to take it. Now, because the cells are unable to take it, the body thinks that there is no glucose in the blood. Therefore, there is this hormone released, which is glucagon. This glucagon further increases the blood glucose levels. Now, can you see, like, it's so much of glucose, right? One was because the glucose itself was not able to enter into skeletal muscle and liver. Okay. Next set of glucose was added by liver by hepatic gluconeogenesis. Next set of glucose was added by glucagon itself. Now there is so much of glucose in the blood. Now this glucose, because now there are two things, right? One, the body is responding because the cells are unable to take the glucose. Other side, now the body will respond because there is lot of glucose in the blood. So high levels of glucose trigger insulin release by the pancreas. Okay. Now this insulin comes to the blood. It ensures the glucose is being taken up by the muscle and the liver. Now we know that there is insulin resistance. Therefore, it's not like all the glucose is entering into these cells. There is still hyperglycemia. High glucose levels are still there, which trigger more insulin release, and this insulin comes again, acts on the receptors, right? So can you see now the body requires now more insulin for the same amount of action, right? So there is this vicious cycle continues until one day what happens is the pancreas actually becomes very tired of releasing more and more insulin. There is finally beta cell failure and there is impaired insulin secretion. So in type 2 what happened was, okay, so this all happened. Blood glucose increased, glucagon was there. Beta cell failure. So initially it started as insulin resistance, but later what happened? There was impaired insulin secretion. So if you see any patient, right, who has diabetes for long period of time, so initially they respond to oral, I'm saying about the type 2 diabetes. Initially they respond to oral hypoglycemic agents, but then what happens? The beta cell failure sets in and there is impaired insulin secretion. So ultimately you will have to start them on insulin. Okay. Now we know that there is lot of glucose in the blood, and we also know that glucose is freely filtered in urine. So all this glucose now what they do? They enter into the urine. Okay. So because of this, glucose is being lost in the urine, therefore there is glucosuria. Now, because glucose is lost, glucose takes with it water also. Therefore, what happens? Water is also being accumulated in the urine, like there is increased water, which is leading to polyuria. Glucoseuria means the patient is actually losing glucose. Whenever the patient is losing glucose, he or she feels very hungry, and they in turn what they do, they compensate it by, the body compensates by making the patient eat more food. So there is polyphagia. Then because the patient is losing water, there is thirst mechanism which are getting activated. The patient drinks more water and there is polydipsia. So these four are the important symptoms: glucosuria, polyuria, because along with glucose water is also lost. Because the glucose is lost, there is polyphagia. Because there is water is lost, there is polydipsia. So these are the important symptoms and these are the important pathophysiology. Like we understood how the insulin was secreted, how the insulin was synthesized, fasting cycle, feeding cycle. So in the fasting cycle, insulin levels are low, glucagon levels are high. In feeding cycle, insulin levels are high and glucagon levels are low. Also, one thing is that, see, in normal individuals, right, postprandially, what we saw, insulin increases, glucagon decreases. But what happens in diabetes? We understood that the body is actually thinking the body doesn't have glucose. It doesn't have glucose. So in diabetic, what happens? Postprandially, insulin is increased, also glucagon is increased. So this is important thing to understand about the fasting feeding cycle. Then we saw about the secretion of insulin, right? ATP, closure of potassium channels, influx of calcium, and then release of insulin. So why does obesity cause diabetes mellitus? Adipocytes, in adipocytes, fat gets accumulated in adipocytes and the muscles. Adipocytes release inflammatory markers. In muscles, what happens? There is impaired insulin receptor signaling. Both of this impair the insulin receptor and cause insulin resistance. Okay. Then we saw about the vicious cycle, like blood glucose levels are there, but like cells are unable to take, therefore the blood glucose are high. Then hepatic gluconeogenesis, increased glucagon, they further increase the glucose. This glucose is lost in the urine, leading to glucosuria, polyuria, polyphagia and polydipsia. I hope this is clear. Okay, and that is it about this video. Thank you.